All right, good afternoon for the last time. Welcome back for the last time. Yeah, you were used to doing this, but everything good comes to an end, including our lives. Remember, you're here for a finite amount of time. Nothing like being morbid, right? So enjoy it. Today we are going to kind of wrap up this course by talking about ways that we can turn viruses to work for us, therapeutic viruses. And you see that quote there by my colleague Dixon Despommier, trust science, not scientists. Very important. We are all human and fallible, but science is not. I was giving a seminar 2 weeks ago at Tulane, and a guy asked me, do you believe in science? I was like, for the first time in my life, I paused many seconds. I didn't know what to say. But actually later, he was a student, a PhD student, and what he meant was, you do experiments and you draw conclusions, but is that science? That's what I think science is. Maybe he didn't think so. That's what we do in science. We have ideas, we want to test them, we design experiments, we get results, we see how they fit. Sometimes we change our hypothesis, sometimes not. And that, by the way, is why many people mistrust scientists, because they think we change our minds all the time. And we do, because we go by the data, right? That's the way it's supposed to be, and many people don't get that. So one of the best things you can do is explain to people that science is a fluid field, and that it's not the scientists that are slippery, but it's the field. It just has to change. Today we are going to talk about 4 different areas where we can take viruses and use them therapeutically. First, we're going to look at phage therapy for bacterial infections. We're going to look at gene therapy, where we give people a gene because they either lack it or have a mutated version. We're going to look at using vectors to deliver antigens, as in viral vaccines. And finally, to kill tumors, oncotherapy. And the key for all of this, let me say it again, the key experiment for all of this is being able to modify a viral genome, whether it be DNA or RNA. RNA genomes can't really be modified. You can't change individual bases readily. You can't cut pieces out and put pieces in, but you can do that with DNA very easily. So the ability to take a DNA copy of either an RNA or a DNA virus, put it in a plasmid, grow it up in bacteria, and then derive virus from that is very important. So this is shown for poliovirus, where we can take the viral RNA and make a DNA copy of it in a plasmid. If you put that DNA into cells, it initiates an infectious cycle. Or if you make RNA transcripts from the plasmid in vitro, the transcripts are also infectious. This is an experiment I did in 1981 as a postdoc. It was the first time anyone had shown that DNA copy of an animal virus genome was infectious, and it was important because it established a principle that this can be done for viruses and it can be useful. And to this day, this technique is used to modify genomes for all the applications that we're going to talk about today. So let's start with phage therapy. Darrell discovered phages in 1915, and he thought they would be useful to treat bacterial infections because on the plates, the agar plates, he saw that they lysed bacteria. He founded the Eliava Institute, which is in Georgia, the country Georgia, not the state of Georgia, and it is active to this day. And in fact, that institute produced phages for antibacterial therapy during World War II. And many soldiers had in their kits a little vial. I have a picture of these vials that have Georgian written on them. Phages to treat intestinal infections. And so for many years, people worked on phage therapy, but the discovery of antimicrobial compounds in the 30s really put a damper on phage therapy because the antimicrobials were very easy to do. Phages, as you will see, are a little bit more difficult to tailor. And so we had many years where we were using just antimicrobials to treat bacterial infections, but now you know we have a lot of antimicrobial resistance globally for many different bacteria. And so this has revitalized our interest in using phages to treat bacterial infections. And there's a huge amount of effort going towards doing this. And I would say if you are interested in viruses and clinical applications, this is a really good area to work in. I think this is going to be a big one. And so the principle is you use a bacteriophage to kill specific bacteria, to lyse them. But of course, you don't want it to lyse your own normal flora. We are full of microbes that are necessary and beneficial for us, so the phage that you give cannot destroy those as well. So you have to, if you have a sick patient, you have to identify the bacterium that's causing the disease. That's not so hard. We have really good ways to do that now. We can determine which phage will lyse it. You have to have a panel of phages ready to be tested. And then, of course, you have to produce it in a manner that is allowed to be put into people. It has to be produced under the right conditions. And so I will give you some examples of using that in patients in a moment. But some other applications of phage therapy include the uses of the proteins encoded in the phage genome that would lyse the bacteria. We call those lysins. Those lysins, as you can see in this picture here, they break the membrane and the peptidoglycan, and they cause the contents of the bacterium to come out, and of course, they end up lysing. These have been approved for surface decontamination, so in areas where you're working with food, you can put these lysins on the surface to decontaminate them, instead of using Mr. Clean, I guess, right? There's another one called AgriPhage, approved in 25, to treat a tomato bacterial disease. It's a phage sprayed onto the plants. And finally, List Shield is a listeriaphage. So listeria are bacteria often associated with foodborne infections, in particular meat and poultry, but others as well. And it's been approved by the FDA for treating these products to get rid of listeria. It's actually the first phage that was given this designation of generally regarded as safe, which is needed to be included into food products. So here are some clinical successes of phage therapy. These are some which I find particularly interesting. First, on the upper left, phage treatment of an aortic graft infected with Pseudomonas aeruginosa. This was a patient who received an aortic graft and it got contaminated, and they developed a phage to treat the infection. It was antimicrobial resistant. They developed a phage to treat it. And if you read the paper, you can get the whole story. The surgeons opened up the patient, and the area of the graft was so broken up, so fragile. They were going to originally inject it into the graft. They took the tube of phage and they just poured it on it and sewed him back up again. He actually recovered and lived until the day the paper was published. He died of something else, unfortunately. The one below it, engineered bacteriophages for treatment of a patient with a disseminated drug-resistant Mycobacterium abscessus. There is a very interesting program called Phages, run by Graham Hatfull and others, where they get high school students to go out in their backyard, get dirt, and isolate Mycobacterium abscessus phages. And they sequence them, they annotate them, they name them after themselves. It's a really cool program. And they used some of those phages to treat this particular patient. And in the course of the infection and treatment, the patient became resistant. The patient's mycobacteria became resistant. They re-engineered the phage to treat it so that you can do this on the fly. Really interesting. On the right, another treatment which made the news, really widespread reporting on this: development and use of personalized bacteriophage-based therapeutic cocktails to treat a patient with a disseminated resistant Acinetobacter baumannii infection. This is a story of a guy who was I think he was in Turkey on vacation. He got really sick. They tried treating him, didn't respond. They airlifted him to Germany. He still didn't respond. Meanwhile, his wife, who's an author on this paper, Stephanie Strathdy, she They're both working at UCSD, and she's looking up phage therapy, and she finds a group in Texas who are doing phage therapy for Acinetobacter. So the end of the story is, at Texas A&M, they grow up phage. They give him intravenous injections for 16 days. He's in a coma. He wakes up, and he's okay after 16 days of intravenous treatment. Interesting story. And we did a podcast down there at Texas A&M talking to some of those individuals. And we also have done, Paul Turner is the author on this paper. He's at Yale, and we did a podcast with him as well, if you want to hear about it. It's very cool. But when you do treat people with phages, you get antibody responses against the phage. So here's a paper that reports that potent antibody-mediated neutralization limits phage treatment. So this is another M. abscessus infection where you get antibodies to the phage arising during treatment, so you're going to have to figure out ways to deal with that. So this is very promising. I don't think it's ready for intravenous application yet, but certainly topical application of infections, it could work well. All right, next I want to talk about using vectors to do different things, and let's talk about the vectors themselves. A number of viruses are typically used as vectors. First, adenoviruses. You guys have all learned about adenoviruses in this course. Double-stranded DNA viruses, icosahedral capsid. The DNA is about 36,000 base pairs of double-stranded linear DNA, and it can take foreign genes, as you will see. The reason these are useful as vectors, they infect post-mitotic cells. What does that mean? They're not undergoing mitosis. And so why can these viruses infect them? Aha, remember E1A, E1B goes in and gets the cells to start dividing. So it doesn't matter that they're post-mitotic. Fast onset of gene expression, the virus remains episomal, it doesn't integrate, so there's no insertional mutagenesis. Has a big capacity. It's easy to make pure concentrated preps. There are a lot of human and animal serotypes. The only drawback is when you give this to someone, they develop immunity. The first generation vectors were deleted for E1 and E3. E1 encodes the T antigens, which are not essential for reproduction. You can take that out and use that space to insert something. And E3 encodes immunomodulatory proteins, so you don't need that either. But this is limited capacity, so people wanted to make bigger capacity vectors. Second generation E1, E3 deletion plus E2 and E4. These were all genes that were deemed not to be essential for vector use through basic research. But the most recent vectors that are used, these are called gutless vectors because you're basically taking out all of the genome except the ends. So we see on the bottom here, there is the whole adenovirus genome, and what's taken out is everything from the left bar to the right bar. And so what the vector consists of is the left-hand inverted terminal repeat, the packaging sequences, and the right-hand inverted terminal repeat. All the genes in the middle are gone, so you have a lot of room for a transgene. And you can stick that in there, and you have your promoter driving it right there. So obviously the packaging sequences are essential to get this DNA packaged into adenoviruses, which is what you use to deliver them to patients. And so one of the ways you can package this, so that's great to have that gutless vector, but how do you get it into a capsid? Well, that is shown here, and it's very clever because you use a helper virus, but you don't want the helper virus to contaminate the preparation, right? So here on the top is vector DNA. I just showed you. It's all deleted except for the packaging sequence and the two ends. You put your transgene in there. So that's the vector DNA. You introduce that into cells that are producing both CRE and E1. So CRE is a recombinase that cuts specifically at these triangular sequences called LOX-P. CRE stands for cream cheese and LOX, and that's true, I'm not lying. That's why they named it. And so if you put LOX-P sites into a DNA and you have CRE endonuclease, it will cut there. And so here's the helper virus at the bottom. This is a full-length adenovirus where the packaging sequences are surrounded by 2 LOX P sites. So we're going to get rid of the packaging sequences when we infect cells with this helper virus. So we take that virus, and that virus is deleted for E1, so it will only reproduce in E1 producing cells. So you take the vector and the helper, you put them into cells that are making Cre in E1. Out comes adenoviruses with only the vector DNA packaged, because the packaging sequence is removed from the helper by the Cre-LOX system. And that DNA, the vector DNA, is packaged because it has a packaging sequence. So the helper provides the capsid proteins, they encapsulate the vector DNA. So really, I tell you this in detail because it's the result of all the molecular biology I've told you about so far in this course. You use that to develop therapeutically useful vectors. Adeno-associated viruses are also extensively used as vectors. Remember, these are rather small icosahedral particles with a single-stranded DNA genome. They encode very few proteins, the capsid proteins and some proteins, the rep protein involved in DNA synthesis. These can be easily modified to take a transgene. So there on the top is the viral genome. So you have 2 inverted terminal repeats. You have the rep coding region, you have the capsid coding region. You can remove both of those. You can put a promoter in and put in a transgene. So now you have a plasmid with a transgene in it. You take that, you take a second plasmid that encodes rep and cap, which you've removed from the transgene encoding plasmid, and then you add other plasmids with helper functions. Remember, this is adenovirus-associated virus. It's a helper-dependent virus. You need to add E1B, E1A, E1B, E2, E4, VA from adenovirus. After all that, out comes recombinant AAV containing your transgene. And the beauty of AAV, you get long-term expression, years worth of expression, and there are many serotypes, and you can make more serotypes by mutating the capsid coding region of these vectors. These viruses persist for years, and that finding was unexpected. No one thought that they would persist. Now, AAV genomes are known to integrate into the host cell genome. They only do so if you infect in the absence of a helper virus. But they require the rep proteins, which are not present in these vectors. So the AAV vectors do not integrate. But what they do is when they're introduced into cells in a capsid, as I've just shown you, they recombine. The DNAs recombine and they make circular monomers and concatemers. And these persist as episomes for many, many years. And that was totally unexpected that this would happen. And obviously it's a benefit because if you need long-term expression, this is the way to go. Now, not every application needs long-term production, right? Maybe an antigen for a vaccine. You don't want to have the antigen expressed forever. But if you're correcting a genetic defect, this could be the way to go. All right, first question is, which technology is indispensable for the production of therapeutic viruses? X-ray crystallography, high-throughput genome sequencing, synthesis of infectious DNA copies of viral genomes, plaque assay, immunofluorescence. Which is indispensable? Okay, let us see how we did. Well, it's 70% is the right answer, synthesis of infectious DNA copies. That's the key. I mean, X-ray crystallography is okay, but it's not indispensable. High throughput sequencing is okay. You don't have to do high throughput. You could do regular genome sequencing. Plaque assay is okay, but if you don't have a plaque assay, which you don't have for every virus, you use other methods. But the DNA is important because it gives you the ability to modify the vector in the ways I've told you and in the ways that we're going to talk about subsequently. Now, retroviruses can also be used as vectors. It's our third vector here. For retroviruses, you start with 2 plasmids. As shown at the top, we have a capsid plasmid and an envelope plasmid. The capsid plasmid just encodes the GAG and the POL proteins with a promoter. The envelope plasmid encodes the spike glycoprotein. When you put both of these into cells, they generate particles, whole retrovirus particles, which are normal, except they don't have a viral genome because there's no genome plasmid here. So these proteins are capable of driving the production of particles. So then if you want to make a retrovirus containing a foreign gene, what you do on the bottom, you have a capsid and an envelope plasmid, and then you include a third plasmid where you have your transgene in it. And there's a promoter there that drives it. And most importantly, this green sequence is the packaging sequence. That's important so that the RNA for the transgene will get packaged into those particles that are assembled by the capsid and the envelope proteins. Among the retroviruses that are used, there are a couple of different retroviruses, but these are the two major ones. On the left, Maloney leukemia virus-based vectors. So these are retroviruses with simple genomes. And then on the right are HIV-based vectors. So we have taken HIV, right, which has infected 37 million people and caused millions of deaths, and we're using it for gene therapy. To me, that just blows me away that we can do that, right? Take a lethal virus and repurpose it. So what we do for both of these viruses, we have made packaging plasmids, which of course contain the structural proteins, and we have envelope plasmids, just like we showed you before. That can be done for both MLV and HIV. And then on the bottom are our transfer plasmids. And I know there are a lot of letters here, but fundamentally it's the same as before. We have a plasmid encoding your transgene, which is the gene of interest. It has a promoter. It has a packaging sequence, the Psi region here. The initial plasmids were made with intact LTRs, and that was problematic because with those we got some insertional mutagenesis in people because the LTRs of course have promoters, and if they sit next to a gene and turn it on, that could be a problem. So on the bottom are the second generation vectors, which are called self-inactivating plasmids. They have deletions in the LTR. So basically, the LTRs are not generated by reverse transcription, and so they are safer after integration into the host. And you'll see an example of where that was a problem in a moment. So these retrovirus vectors, again, based on HIV and others. HIV is interesting because it can infect non-dividing cells, and many of the cells you want to target are not dividing, so that's important. These viruses, of course, integrate, which is a benefit because it's long-term expression. You can put reasonably big inserts into them, but of course, you can get insertional mutagenesis. And so, we didn't know that until we did the first round of gene therapy, and some people got cancers from the vectors, so then we had to modify them to make them either integration deficient, which takes away one of the major benefits, or inactivate the LTR, so it doesn't have a promoter in it. And the final thing that's very good here is that you don't have to use the retrovirus glycoprotein when you make these particles. You could use others from other viruses, and a common one is the VSV, vesicular stomatitis virus G, or glycoprotein, which will insert into these retroviruses. That's called pseudotyping, when you put a different spike protein on. So why would you do that? Because G has a broad host range. It can infect a lot of different cell types. Whereas, as you know, the spike of HIV-1 will only infect CD4 positive cells. So if you want to change the host range, you can do that. Pox viruses can also be used as vectors. In fact, the vector that we use is called modified vaccinia virus Ankara. The vaccine that we used to use, or we still use in certain populations for smallpox, was derived from vaccinia virus. And this one is a modified version of it, which was made as an alternative because the original smallpox vaccine caused a lot of side effects. And so attempts were made to get rid of those, and this virus was produced as an alternative. It is now part of the National Strategic Stockpile of the US. Did you know we have stockpile of MVA and 2 antivirals in case there is a smallpox bioterrorism attack? Because no one, or very few people, are immunized anymore against smallpox except the military. And so someone could introduce the virus and start a bioterror attack. So we have vaccines and antivirals ready. And they were helpful, right, during the Mpox outbreak, because we broke into the stockpile and used the vaccine and the antiviral. This is a replication defective vector. It was made by passaging the original vaccine in chicken cells until it was well adapted to chicken cells. It's infectious in chicken cells or bird cells in general, but not in mammalian cells. There's an assembly block. So the virus will get into your cells, it will do gene expression, but it won't make new virus particles. So perfect for a vaccine. You can work with this under BSL-1. High school laboratories can work with this vector. BSL-1 is just very, very limited containment, has big capacity, and we also have other poxvirus vectors, including ones derived from canary pox viruses. Yes, there are pox viruses that infect birds. The way you make vectors with this virus is very interesting. You have your MVA virus right there at the top, and you infect cells with it, and then you have your transgene. So you have some transgene, which is shown here in blue, and you want to get it into the vaccinia virus, or the MVA virus. So what you have done is introduce into this plasmid some sequences derived from the MVA genome, and they're going to direct homologous recombination between the transgene vector and MVA. So you transfect this transgene into the cells that are infected with MVA. The DNA of the virus and this DNA will recombine, and they do so at very high frequency, and out come recombinants. You just need to identify them in some way, which isn't very hard to do. You can use vesicular stomatitis virus as a vector. This is a virus with a negative strand RNA genome. Of course, we make DNA copy of it so we can modify it. Typically, we replace the glycoprotein coding region with a transgene of choice. And then to make virus, we have plasmids. We do a 4-plasmid transfection with the NPL genes on plasmids, and then you have the genome, the RNA genome of the virus with your transgene in it. You put those into cells. Each of these plasmids has a promoter for T7 RNA polymerase, which you introduce separately by a vaccinia virus encoding the polymerase. And so 4 plasmids, vaccinia virus producing T7, out comes infectious vesicular stomatitis virus. Now, of course, in this case, you're going to want to put something in it that will allow this virus to attach to cells. So it's going to be typically another glycoprotein of another virus. Flaviviruses, plus-stranded RNA viruses, yellow fever, dengue, West Nile virus, these have been derived as vectors as well. In particular, we have a yellow fever vaccine that has been used for many years. And so what was done was to clone the genome of that vaccine virus as a DNA, and then replace the structural proteins with those of dengue virus. And instantly you have a dengue virus vaccine because the yellow fever backbone provides all of the attenuating changes, the ones that prevent the virus from causing disease. And so to make virus, you take that DNA, again, yellow fever vaccine with dengue virus structural proteins, you make RNA in vitro, you put that into cells, and you get your vaccine virus out. And there is a vaccine for dengue, Dengvaxia, for serotypes, which was made using this approach. Alpha viruses, these are the plus-stranded RNA viruses that have a subgenomic mRNA. You can also use these to vector related viral structural proteins. You replace the structural protein genes with another alpha virus that's of medical relevance. You do that in a DNA copy of the genome. You make RNA transcripts in vitro, transfect those, and you have your vaccine viruses. And one more, Newcastle disease virus. This is a negative strand RNA virus, very much like VSV, but a different family, paramyxoviruses. So think measles. This is a virus of chickens. Farmers vaccinate their chickens against Newcastle disease virus because it's quite deadly. It has been modified to be a vector, and typically, so there's a DNA copy, of course, of the RNA genome. You can insert your transgene here between the P and the M coding region, so you're adding an additional gene in there. And again, you do a 4-plasmid transfection into cells infected with vaccinia-producing T7 RNA polymerase and out come viruses. And again, just to clarify, the T7 RNA polymerase produced by vaccinia virus drives the promoter on each of these plasmids, and that gives you transcripts which initiate the infectious cycle. So that's just a quick run-through of some of the vectors that we use for vaccines. And here's a list of licensed vaccines that use some of these viral vectors. All right, these are not traditional vaccines. These are viral vectored vaccines. So Ervebo is the Zaire Ebola virus glycoprotein in a VSV vector. So that's licensed. Dengvaxia, as I told you, is the structural region of the 4 dengue serotypes in a yellow fever vaccine vector. This same yellow fever vaccine vector has been used for structural regions of Japanese encephalitis virus. This is not a virus we have here, but it's present in other countries, and so they vaccinate against it, and if you're going there, you should be vaccinated against it as well. And there's also a West Nile virus vaccine for horses, which is again vectored in the yellow fever vaccine vectored backbone. There is, as you may know, avian H5 influenza virus, H5N1 influenza virus, is a big threat to domestic farmed birds, chickens, and geese, and so forth. So there is a vaccine against H5 avian influenza virus, which is vectored in a Newcastle disease virus vector. So the farmers can give this to their chickens and immunize them against both Newcastle disease virus and H5N1 influenza virus as well. And this is important. So I was just at the University of New Hampshire last week talking with a veterinary microbiologist, and he said the farms all around him with chickens are having these outbreaks of high pathogenic avian H5N1 influenza virus. I had one farm with just a few chickens, 100 chickens, all outdoors. The farmer called them and said, our chickens are sick. So they said, okay, we'll send someone to swab them. By the time they got there, the chickens were all dead of avian H5 influenza virus. And what happens is they had a little pond on their farm, and the wild ducks come, and they come into the pond, they excrete avian H5N1 influenza virus, and the chickens get it. So really, if you keep your chickens outside, it's a problem. You have to keep them indoors. Of course, one of the COVID vaccines made by Janssen is vectored in an adenovirus 26 vector, 26 serotype. The type 26 is rare in humans, so there's not likely to be much antibody against it. That's the one that also caused blood clotting, if you remember, and we don't really use that anymore in the US. The Chadox is an adenovirus vector. Again, is licensed for COVID. It's called Vaxzevria. It's the spike protein of, as is adeno26, the spike protein gene of SARS-CoV-2 in a chimpanzee adenovirus vector. And they use that because no humans are infected with that particular serotype. And finally, in Russia, they made a COVID vaccine which they call Sputnik. Do you know why they called it Sputnik? Do you know what Sputnik was in the first place? Yeah.
Is it because the adenovirus is kind of looks like?
It looks like a Sputnik? No. Sputnik was the first satellite to go up. So they said this was the first COVID vaccine, so they called it Sputnik. That's kind of not even clear it was the first. Anyway, this is a 2-shot vaccine. Add 5 first and add 26 second. So if you need 2, it's best to have the second one a different serotype, right? So that's what they did there. All right. Let's talk about gene therapy. This is done for what's called monogenic diseases. It's caused by a mutation in one gene. There are over 6,000 different human monogenic diseases, and 1 out of 200 live births is a kid with a monogenic disease. Very common, so this is really applicable work. So you have a defect in a gene. Here we have a diagram of that. We have a defect in a gene which you can track the inheritance of, you can think about replacing it with a viral vector. And currently there are over 1,800 clinical trials in progress to test viral gene therapy for such diseases. Here are just a few of them. Obviously there are many, many more. Different kinds of diseases caused by defects in one gene, and this table gives you the incidence and the vector that is being used to try and take care of these. For example, SCID is severe combined immunodeficiency. This is a particularly bad one where the defect can be one of two different defects in the ADA gene or the common cytokine receptor gamma chain. You don't have any B cells or T cells or NK cells. It's a big problem. So people are trying to deliver these genes to those individuals with retrovirus vectors. Hemophilia is another one. The hemoglobinopathies are others. The retinal degenerative diseases that cause blindness we'll talk about. That's been successfully taken care of with AAV vectors and others as well. But obviously many, many more. The number of trials for gene therapy have gone up, obviously, since the 80s when we got the ability to modify viral genomes. And the different kinds of indications that are addressed are shown here on the left. Cancer, monogenic diseases, which is what we're talking about here, also other things like infectious diseases, cardiovascular, neurological, and so forth. And on the right are the various vectors that are used. Adenovirus is a big one, retroviruses, adeno-associated virus there, and even naked plasmid DNA in some cases. The way these work, there are two distinct ways of giving a patient a gene. So first you can just put the virus into them. So on the top, that's called direct delivery, where you've taken the gene of interest, you've cloned it into your vector, and you simply infuse that into the patient. The drawback of that is if you put it intravenously, it will first go to the liver, and most of it gets cleared in the liver. And so if you're targeting a liver disease, that's great, but if you're targeting other diseases, it's not so great. The alternative is to do what we call cell-based delivery, which we will talk a bit about later. For example, you can take T cells from a patient, so you would take the blood from a patient, take out the T cells, and introduce into them a chimeric antigen receptor, which will now give the T cells the ability to target an antigen, say, on a tumor cell. And those will go in and kill the tumor or lyse the tumor. Or you can take stem cells out of the patient, or you can take stem cells from a cell bank or from embryonic stem cells. You can then introduce the gene of interest into them and reinfuse them into the patient. And because they're stem cells, they'll give rise to a variety of other tissues and propagate the gene as well. So that's a little bit more effective, a lot more effective than just injecting the virus. And the T cell is an example where you absolutely know what specific cell type you want to put the gene in. So some of the early viral gene therapy trials, 1993 was an early one. Cystic fibrosis was a disease targeted early. This is, of course, a lung disease where patients are lacking or have a mutation in the cystic fibrosis transmembrane conductance regulator, and it predisposes them to severe lung infections with Pseudomonas, for example. So the idea is, let's give them the wild type gene. So this very first trial was done with a 23-year-old male. That's him lying there. And with cystic fibrosis, he was homozygous for a mutation in the CFTR gene. So they decided to clone the wild type CFTR gene into a first generation adenovirus vector, and they sprayed 2 × 10 to the 8th PFU directly into his lung. You see they put a hose right down into his lung and sprayed the virus in there, and they sprayed it down into his lung. And then they measured the production of the CFTR protein. And pre-therapy, here are some of his lung cells stained for the protein with antibodies. And 4 days post-therapy, you can see they're making the protein. And if you graph that, here's exogenous over endogenous synthesis. You can see a nice peak of protein production a few days after infusion, and then that goes down. because these are acute virus infections, right? Anyway, they give them another infusion. Sometime later, you get another burst. And then the third infusion, you get nothing. And that's because now the patient's made antibodies to the vector, and so you no longer get expression. So this doesn't provide any long-term benefit. In fact, right now, there's no licensed gene therapy for this disease, even though it's one that's obviously fit for this, because none of these have worked, and other approaches haven't worked either. I think people are now looking into CRISPR, using CRISPR to try and repair the mutation, but CRISPR has got its own problems, has off-target effects that people haven't learned to deal with yet. So that's going to be some time off. Another early one was an attempt to treat a disease called ornithine transcarbamylase deficiency. It's an X-linked disease that leads to accumulation of ammonia and glutamate in the blood. And if you have severe versions of this disease, you have issues, declining cognitive ability, and you can die. So the trial was done at the University of Pennsylvania. A number of patients who were recruited, including this young man, Jesse Gelsinger, who was a teenager, and he had a mild version of the disease, but he and his dad thought it would be good to participate in this. So he was given an adenovirus vector with a wild-type OTC gene. 4 days later, he died. Massive immune response, multiple organ failure. And it turns out, in retrospect, they broke a lot of rules at Penn just so they could get this trial done. They overlooked a lot of the preclinical non-human primate data, which said there were severe reactions in the non-human primates. And they shouldn't have done this, essentially. So if you go online, you can find lots of writings by his dad about this very sad situation. And so because of this, we took a step backwards and reassessed all of our gene therapy approaches. Another example of problems, attempts to cure X-linked severe combined immunodeficiency. As I said, this is a defect in one of two genes, and this one, the IL-2 receptor gamma chain, leading to absence of TB and K cells. And that map just shows you, this is the IL-2R gamma chain gene, and these dots are mutations in different patients that affect the function of that gene. So the idea is to give patients back the normal gene. So there were two trials initiated in London and Paris, and this is done with infants because you don't want to grow up with SCID because you can't go out. You get infections all the time. So you want to treat them as infants. So they take out bone marrow hematopoietic precursor cells, CD34 positive. They put them in culture. They transduce them with a retrovirus vector that has the wild-type gene. They make sure everything is good with the cells. They put them back in the patients. Now they will colonize the bone marrow and begin making normal TB and K cells. And this actually worked, except that some of the patients, some of the babies, developed T-cell leukemia 3 to 6 years after treatment. And so there were something like 27 trials ongoing with retroviral vectors, so they were all stopped, and these were studied very intensively. And it turns out that in these kids who developed leukemias, the vector inserted near a known oncogene. Remember, oncogenes are genes encoding proteins involved in cell growth control, and they shouldn't be on when they're not supposed to be. But when you have a retrovirus integrating near the oncogene, here's the promoter for the oncogene here. They turn on the oncogene, and that's what leads to the oncogenesis. And so because of that, the vectors were modified so they no longer had a right-hand LTR, and they would no longer do this. And so subsequently, a successful trial was done again with infants with SCID. They gave them lentiviral gene therapy with bone marrow transplants, and after 18 months, all these kids had functional B and T cells. So this is a very nice success story showing that this can work, and you can get around limitations as long as you know the molecular biology, which again you get from fundamental science. None of them died. The leukemia actually was treated by chemotherapy and so forth, so that was good. But you don't want to have that as a side effect, right? So this is better. Sickle cell disease is another one you can treat by gene therapy. So sickle cell, of course, you have a mutated beta globin gene that makes altered protein that forms fibrils, makes aberrant red blood cells. So you want to replace the gene. And so you could put the gene in a lentiviral vector, the wild type gene, and deliver that to patients. That certainly works. But more recently, people have explored using CRISPR-mediated gene replacement. You deliver the CRISPR system with viral vectors, and you basically are recombining out the mutations in the gene with the wild type version. And so some of these are getting close to FDA approval for treating sickle cell disease. Now, the one I really like is treatment of inherited retinopathies. These are blinding conditions. They're inherited, right? So you inherit a mutation in the gene and you're blind. They're monogenic. And the genes, there are many genes that can be involved. They are in the retinal photoreceptors and the pigment epithelium. So remember, at the back of your eye is the retina, and in a close-up here, we have a look at the neuronal construction of the retina. We have the retinal pigment epithelium, which receives the light, and then a series of neurons below it that transduce the signal to the brain. And there can be defects in the photoreceptors themselves, or the neuronal photoreceptors, or the retinal pigment epithelium themselves. And this table on the right shows you the different genes, the mutations in different genes that are involved in the RPE, the rods and the cones, photoreceptor rods and cones, and these cells below them, these neuronal cells. And so in each case, it's a different gene that has a mutation, but they all lead to a blinding condition. And so very many trials have been done using AAV, lentiviral and adenovirus vectors to supply each of these genes to the patients who have the defect. And one of them has really worked. It's led to an FDA-approved drug. It's called Leber congenital amaurosis. It has mutations in the RPE65 gene. Is that here? It's going to be up here in the RPE, one of these genes here. So they deliver that. They put it in an AAV vector. It was initially tested in dogs with dogs to make sure it restored their vision. And it restored it for many years, right? Because it gives long-term expression, as I told you, for these AAV vectors. And then it was tested in humans and approved by the FDA in 2017. So the drug is called Luxturna. It can be used to treat both eyes or one eye. And the way it's done is it's injected into the retinal pigment epithelium. So they put a needle through the eyeball into the RPE, inject the vector into there. And many of these people see for the first time in their lives. You can search videos on YouTube where they, say very touching things. Anyway, Catherine High used to be at the University of Pennsylvania. She was involved in the development of that, and I interviewed her some time ago. Really interesting chat with her. So these are the successes that I've described, and also some I haven't. We have SCID, adenosine deaminase deficiency, Leber congenital amaurosis. Luxturna, by the way, $895,000 for 2 eyes. It's a lot of money, right? If you ask the company why it's so expensive, do you know what they will tell you? How badly do you want to see? And so, this is something that the insurance companies have to deal with, whether they're going to pay for it or not. Hemophilia, beta thalassemia, lipoprotein lipase, and then at the bottom, the most expensive drug ever is an AAV carrying the spinal motor neuron 1 gene for biallelic spinal muscular atrophy. $2.25 million, most expensive drug ever. It's one shot, right? How much do you want to not develop this disease? It's kind of obnoxious for them to say it that way, but that's what it's based on, because it's not just about recovering their development costs. Obviously, they're making far more than that, but that's a whole nother story that it's a whole topic for another course, actually, the price of drugs. And who else? I mean, we're talking about in the US, people not affording this. What about other countries? My philosophy is that everyone in the world should get the results of research, but unfortunately it doesn't happen. Next question. Which of these viral vectors are not likely to be compromised by immune memory in humans? Newcastle disease virus, vaccinia virus, herpes simplex virus, adenovirus. So which one you're not likely to have immune memory? Why? What do I have to update it?
Most expensive drug.
Oh, what's the most expensive one?
Lentiviral treatment for adrenal leukodystrophy, approved late last year.
Yeah, I used to talk about that in this course, adrenal leukodystrophy. Yeah, let's check it out. Adrenal.
Like this, nice.
That was first initiated at Columbia years ago. ALD, yeah. So what's the gene therapy? Oh, they say Luxturna here is, oh, look at this. It's eye-popping. You get it? It's eye-popping? All right, but we don't want to know that. $3 million per treatment. There you go. Bluebird. Do you have stock in this, Richard? Okay, thank you. Got to change this slide for next year. Here we go. I used to talk about adrenal leukodystrophy in this course because some people at Columbia were involved in the initial trials, like Art Banks and so forth. Oh, interesting. 3 million. It's really an interesting issue, right? How much a drug should cost. Okay, what do we have here? Newcastle disease virus is the answer. No, humans are not likely to be infected with that, right? It's not a human virus, but vaccinia is, herpes simplex is, and adenovirus is. So that's why that is the answer. Okay, let's talk about viral oncotherapy, destroying viruses, destroying tumors with viruses. I think this is great. This is going to treat most cancers in 10 years. You heard it here. And if you want to do cool things with viruses and clinical applications, work on this. So the application is we have some viruses that are from animals, non-human viruses that selectively replicate in human tumors. And myxoma, the one they used in Australia to get rid of rabbits, that's one of them. Seneca Valley virus is another. And then we can modify viruses to treat tumors, and sometimes we put immune modulating genes in them. We'll talk about that today. I have a video here by Linda Coughlin who explains this in a nice way. Now, but in the ancient days, the 40s and 50s, people tried to kill tumors with viruses. So here are 4 trials for Hodgkin's, various cancers, acute leukemia, cervical carcinoma, where they just gave people, look at that one, 1949, hepatitis virus. They didn't even know which one it was. Of course, 14 of the patients got hepatitis, and one of them died. And the next one, they used West Nile virus. And some of them got encephalitis. Remember, that's an issue with West Nile virus. The 1953 leukemia, Epstein-Barr virus, yeah, they got mono. And finally, adenovirus. See, with all of them, transient tumor necrosis. So that suggested something might work, but the trials were just horrible. We'd never allow these to happen today. There were no controls. The source of the virus and the concentration, the route varied from patient to patient within a trial. We didn't know how much virus was present. So none of these yielded any useful information. But I would say there was transient response which said maybe we could use viruses to treat tumors. So fast forward today, we have sophisticated ways to modify viruses to be specific for tumors. And I want to remind you that many viruses encode antagonists of immune responses. We talked about that a long time ago, including interferon responses, right? So it turns out that many tumors have defects in the interferon pathway. So why is that of use for designing viruses to kill the tumors? Well, if you give someone a virus to kill a tumor, it may infect other cells And because it antagonizes innate immune responses, it could get out of control, right? So what you can do is take out those genes from the virus because you don't need them to infect the tumor. The tumor is interferon defective already. So that's really cool. So here's some example of 4 different viruses: adenovirus, herpes, vaccinia, MVA, and VSV, where you can mutate different genes that will and get rid of the antagonizing of the interferon response. And that makes it specific for the tumor then, because these viruses are going to be eliminated by the interferon response in other tissues. So that's really good. It's a nice application of having studied for many years how these different viruses antagonize interferon responses. So that's just one thing that we can do to modify these viruses. We can modify viruses to target tumors by the receptor. So for example, tumors often produce new proteins called neoantigens. Remember, they're highly mutated because they're just dividing forever, so they have lots of mutations in different genes. And some of those produce novel antigens. We can identify those, and we can alter viruses so that they bind those specifically. So measles has been modified to recognize a tumor-specific antigen. The herpes glycoprotein has been engineered to recognize proteins that are on gliomas and breast tumors. Adenoviruses have also been modified in the same way. So you can make a oncolytic virus tumor-specific at that attachment step because tumors have unique antigens. You can also make specificity after entry. This is called post-entry targeting. So 2 different ways, positive or negative. Positive means the virus will only work in a tumor cell. So here we use a promoter that's inactive in a healthy cell. So the healthy cell is on the left there. Your therapeutic virus gets in, the promoter doesn't work, so there's no virus reproduction. And you can identify such promoters pretty easily. And then that same promoter works in a tumor cell, so the virus gets into the tumor cell, reproduces, and kills the tumor cell. So that's positive targeting. Negative targeting is where something doesn't happen in a tumor cell to allow the virus to reproduce. So what you do is you put in the vector genome the target for a microRNA that's only present in normal non-tumor cells. So remember, microRNAs will bind to RNAs and degrade them and inactivate them. So on the left here in the healthy cell, the microRNA is present, so it inactivates the virus vector, so it's not going to be reproducing in those cells. And you pick the microRNA target so that the microRNA is not present in tumor cells, so the vector will reproduce. And it's easy to do this. On the right is a Western blot showing the production of 2 different microRNAs in different tissues. And you can see you can easily find microRNAs that are tissue specific, and you use those. All right, so that's an example of negative targeting. We can also put enzymes into these vectors that will activate a drug that you deliver along with the vector. It's hard to infect 100% of the cells in a tumor, but if the tumor cells that are infected make a drug that will kill the surrounding cells, that can be useful. It's called bystander killing. And so one of the things that we do is put the genes for prodrug convertases into these vectors. Those are like thymidine kinase, which converts acyclovir into acyclovir monophosphate, so it will only happen in the cell that's infected with the oncolytic virus. And that acyclovir will eventually be triphosphorylated and inactivate replication. We can also put other genes encoding ion transport proteins that you can then treat the cells or the patient with ionizing radiation. This will help that enter the cell. And immunostimulatory factors, various cytokines, can be included in the vector, which will then be produced and attract the immune system to the tumor. Because typically the tumor is being ignored, and this way you can attract the immune system and help it destroy the tumor. Another example of an oncolytic virus is myxoma virus. So here's an animal virus that was used to kill European rabbits in Australia. It doesn't replicate in any non-rabbit host, but it does infect many types of human cancer cells. So this is very typical. You can find animal viruses that don't infect humans, but human tumor cells will be infected. And why is that? Well, sometimes these tumor cells have poor antiviral responses. Sometimes, because they're tumor cells, remember they're transformed, there are certain cell pathways that are activated, and for reasons that are not fully understood, that allows viruses to reproduce in them. So myxoma virus is used in a situation where you have a patient with a tumor or a cancer that's been refractory to treatments. And let's say you want to take out their bone marrow and then give them lethal chemotherapy or radiation to kill all the tumor cells in them. And the reason you take the bone marrow out is because you're going to kill that too, and you want to give them back some bone marrow so that they can live. The problem is the bone marrow may have tumor cells in it. So we use myxoma virus to kill them. And then you can put them back. You can confirm there's no tumor cells there, and you can put them back in the patient. And these are just a number of studies with different kinds of cancer, usually in mouse models where we engraft the human cancer into the mouse. And before we put the tumor in the mouse, we treat it with myxoma virus, and then we say what happens in the mice. And you can see sometimes 90% of the mice are free of cancer cells, sometimes 100% of them, 100%. So again, you treat the tumor with myxoma virus to kill it and see the effect. And the reason for this, as I said, is so that we can lethally irradiate the individual and get rid of the cancers. Measles virus is also being tested for oncolytic purposes. And interestingly, it's the vaccine strain of measles virus which preferentially replicates in tumors. And one of the properties of the vaccine strain is it can't antagonize two members of interferon signaling pathways, STAT1 and MDA5. And so it preferentially replicates in tumors, which are defective in those, and normal cells are not. This virus was added to this virus the gene for a transporter, a sodium iodide symporter. And during therapy of patients, they give them gamma-emitting isotopes to visualize the tumor, and they can give them beta-emitting isotopes to poison the tumor, and those all get in through this symporter into the tumor cell, right? So that's kind of illustrated there. We have a tumor cell being infected with this measles virus vector. It produces the sodium iodide symporter, which is simply a channel through which these ions can go. You then give ionizing radiation to the patients, and they get into the cell and help kill it. And so one trial was done with two patients who had multiple myeloma. This is a tumor of B cells, where your B cells are clonal. And you can see these are fax plots of cells from these patients. And these are plasmacytoma cells, and antibody-producing cells. And you can see that both patients had this is pre-treatment, normal and clonal PCs, which are the tumor cells. And this one only had clonal plasmacytoma cells, so more extensive propagation. And they were given measles virus vaccine modified in a way that I've just told you, intravenously. And all of their clonal PCs went away, as you can see here. But this is post-treatment, this panel. No more clonal PCs there and there. So the virus kills all of the clonal plasmacytoma cells. In addition, this particular patient had a tumor on her skull. You can see the bulge there. And post-treatment, it was gone. These people were given intravenously 10 to the 11th particles. And one of them said she could feel the pressure on her skull releasing with hours after treatment, because this is a rapidly multiplying virus, as you know. Another virus vector for tumor that's actually been FDA approved is for treating melanoma. Thank you, melanoma. And so this is a vector called telimogene laherparivec, or TVEC. It's herpes virus, which has a gene for GM-CSF, and that will recruit cells to the tumor. We delete 2 genes of the virus, ICP34.5, US11, making the virus reproduce specifically in the tumors. ICP47 is a viral gene encoding an antigen presentation inhibitor. So now we have no inhibition of antigen presentation, so the tumor cells are going to be recognized by cytotoxic T cells. This went through phase 3 for melanoma, which, as you know, is quite diffuse and not amenable to surgery, and it get a good response, and so it was approved in 2015 for treatment of melanomas. Another vaccinia virus is called JX594. This company is called Generex, by the way, I guess in honor of Edward Jenner. This is a virus, again, with GM-CSF, deletion of the TK gene, which makes it more specific for tumors. And this one is tested specifically for, let's say you have a patient with lots of metastases where you can't inject the virus intratumorally. In fact, the previous one, the herpes virus, this was injected into the melanoma and gave a good response. But often you have so many tumors you can't do it. So could we put these viruses intravenously, and would they hit tumors throughout the patient? So they treated 23 patients with all sorts of different tumors. You can see lung, colorectal, melanoma, et cetera. All of these were treatment refractory, so this is a last resort. And the virus replicated in tumors in about half of the patients. Half of them had anti-tumor activity. And you can see these are rectal, endometrial, and colon tissue samples. And you can see the green is the virus reproducing in them. So it's a proof of principle that you can intravenously deliver these viruses, and they will make it to the tumors. And again, we can put prodrug convertases in these vectors, thymidine kinase, cytosine deaminase. They will make chain terminators that stop DNA replication in tumor cells, and one of these vectors is called TOCA511, which is a murine retrovirus with cytosine deaminase. You give the virus either right in the tumor or intravenously. You combine it with fluorocytosine, which is converted by the gene in the vector to 5-FU, and that helps to kill the tumor. And finally, even poliovirus has been repurposed to kill gliomas. This is the Sabin strain, where the iris has been replaced from a rhinovirus. This attenuates the virus, and tumor cells happen to upregulate the polio receptor. So this was given to patients with glioma who you know have a poor prognosis intratumorally, and the median survival time was extended from 12.5 to 11.3 months. So this is being pursued as a potential therapy because there's really nothing else to be done. And I want to just mention in closing that when you use these vectors to kill tumors, you're not just killing the tumor with the virus, that's part of it, but you're releasing antigens from the killed tumor cells, and that attracts antigen-presenting cells like macrophages and dendritic cells. Many of these vectors include cytokine genes that attract tumor cells. So you're really amplifying the immune response. So the killing of the tumor is a combination of the virus and the immune response. Finally, cancer immunotherapy using CAR T cells. I mentioned this earlier. Many tumors have unique antigens, but for some reason our T cells are not recognizing them. So we can make what are called chimeric antigen receptors, where we make a single chain antibody that recognizes this tumor antigen, and then we combine it with other elements, deliver them to patients, so the T cells are harvested from the patient, and then we infect them with a lentivirus vector that expresses this chimeric antigen receptor. And these T cells will now produce the protein and recognize the tumor cell and kill them. And this is being tested for a variety of tumors. It's also being tested using macrophages and NK cells to deliver the CAR receptor. So all of these approaches are, to me, looking really promising for cancer immunotherapy. So all of this today is really a result of basic research. We have advances in virology, recombinant DNA, immunology, clinical science, all of which you have learned in this course. And part of it is luck, dumb luck, right? Serendipity. Sometimes you're just curious about something and you find it and it turns out to be incredibly powerful. So I want to leave you with this thought that you can't always say, I want to cure cancer. You have to let scientists follow their curiosity. I'm curious about how this works. So there has to be a balance between what I call translational research and basic research, and that's what brings us to the best output. All right, here's day one, folks. This is the end. Day one, you didn't know any virology. Now you know more virology than most of the world. I want to thank you for taking this course. And more importantly, don't forget what you've learned. I mean, a lot of the specific knowledge will fade away because you don't use it, but you know where it is now, and you know where to find me if you need some help. And in fact, here are all the places I am on social media, my multiple podcasts, and my blog, Microbe TV, Twitter, Instagram, YouTube, Facebook. Follow me somewhere, keep in touch, and that's my license plate, yes. Thank you so much.